Today we will look at another important process that occur in our cells. Think that at the time when you born, will you have the same number of cells when you become adult? Obviously not. The number of cells in our body increase as we grow up. So what happens in our body that help to increase the number of cells in our body? This is division of a cells. The division phase may consist of mitosis or meiosis, depending on the cell type. If you still not subscribed my channel, hit that subscribe button right now and click the bell icon. So let's discuss this amazing topic which is called mitosis. Mitosis is observed by Walter Fleming. He noticed that during division of a cell, the nucleus passes through a series of events, which he called mitosis. In the mitosis, the cell divides into two daughter cells and the chromosome number remains the same in both of the daughter cells. Mitosis occurs in somatic cells. Somatic cells are the body cells which do not include the reproductive cells. Look at this figure. In this figure, the parent cell divides into two daughter cells and the number of chromosomes in the parent cell is 2n, while the chromosome number remains the same in both of the daughter cells. And this diagram shows the process of mitosis. To understand the process of mitosis better and easily, it is divided into two stages. The first one is karyokinesis and the second one is cytokinesis. Karyokinesis, karyo mean nucleus and kinesis mean division. Karyokinesis is again divided into its phases, in which the first one is prophase, metaphase, anaphase, and the last one is telophase. We will look at each of these in detail. The first one phase in karyokinesis is prophase. In prophase, the chromatin network condenses and forms chromosomes. The centrosomes also move to the opposite poles of the cell in this phase. And in the same time, the formation of spindle fibers also start, while the nuclear envelope and the nucleus present in the cytoplasm of the cells also disappear from the cell in this phase. The second one phase of karyokinesis is metaphase. In this phase, the chromosomes arrange themselves on the metaphase plate. Metaphase plate is at the middle of the cells in which the chromosomes arrange themselves. It is also called equatorial plate. The next event occurs in metaphase is that the spindle fibers attached to each chromosomes from both sides mean the two spindle fibers attached to one chromosome from both sides at the area of kinetochore. You might thinking about that what microtubules or spindle fiber is. So let me shortly explain it. Microtubules are cytoskeleton structure which is present in eukaryotic cell and formed during a cell division to separate the sister chromatid from each other. The main microtubules are astral microtubules, polar microtubules, and kinetochore microtubules. Astral microtubules, those microtubules which originate from the centrosomes and does not connect to the chromosomes, are called astral microtubules, while some are the microtubules that originate from the centrosome and does not connect to the chromosome, but they connect to the another one microtubules which originated from the opposite centrosomes and are called polar microtubules. And the last one is that kinetochore microtubules. These microtubules begins from the centrosomes and connect to the chromosomes kinetochore. These are the microtubules which separate these sister chromatids. The third one phase is anaphase. In this phase, the chromosome splits and move towards the opposite poles. In this phase, these chromosomes which are attached to each other and are called sister chromatids, are split at the kinetochore area and moves towards the opposite poles. Once the chromatid splits, they are called daughter chromosomes. And when these chromatids split from each other, separate from each other by spindle fibers, then these two are called daughter chromosomes, and these two also called daughter chromosomes. The centrosomes begins to move backward to pull these chromatids from each other to the opposite poles. The last one is telophase. In this phase, the karyokinesis division will be completed, means the nucleus division will be completed. It is the reverse of prophase. It means that what happened in the prophase will reversely happen in telophase. The first one, even that happened in telophase, is chromosome reach the opposite poles. The chromosomes that start moving towards the opposite poles in anaphase will reach to the opposite poles in telophase. The second event that occurs is the nuclear envelope formed around the chromosomes. The nuclear envelope will again start reappearing around the chromosomes. The nucleolus will also reappear in telophase, which has disappeared in prophase. The chromosome begins to decondense back into chromatin network, and the cell membranes begins to move inward at the center of the cell. With the end of telophase, we come to our next stage of mitosis, which is called cytokinesis. Cyto mean cytoplasm and kinesis mean division. After the nucleus division, the cytoplasm also divide, which is called cytokinesis. The cytokinesis in plants is a little bit different from the animals' cytokinesis. In animals, the cleavage appears between the cytoplasm of two nuclei. The cell membrane begins to move inward in telophase and deepen and finally produce two identical cells. But there is no such thing happen in plant cells. In plant cells, the cell plate is formed between the two daughter nuclei and divide the cell into two daughter cells. The cell plate is formed by the fusion of small fluid-filled vesicles that produced by Golgi body. These vesicles deposit and fuse together here and form a cell plate, which divide these two daughter nuclei into two daughter cells. This will form a cell plate and divide these two daughter nuclei into two daughter cells.